Good morning, dear students. Today we are going to discuss about the concept of gene. Now, what is a gene? A gene is the basic physical and functional unit of hereditary. Okay. Genes are made up of DNA, right? Rather, you can say they are polynucleotide sequences, right? A gene is a segment of DNA, right? Now, some genes act as instructions to make molecules which are called as proteins. Rather, we can say that genes direct the synthesis of proteins or enzymes. However, there are genes also which do not code for any protein. Slowly we'll discuss about everything. Now, in human beings, genes vary in size from a few hundred DNA bases to as many as 2 million bases. That means gene can be a small segment of DNA or it can be a long, very long segment of DNA. So we said it may be very small in size, measuring few hundred DNA bases maybe up to 2 million bases. Now the Human Gene Project has estimated that humans have between about like 20,000 to 25,000 genes. Okay. Now, here today we are going to discuss about what is a gene, what are the various units of gene, especially what is a cistron, what is a muton, and what is a recon. Now, genes, again, as we said, they are made up of DNA, and each chromosome contains many genes. Okay, each chromosome contains many genes. So, in this diagram, we have shown this is a chromosome, okay, and each chromosome contains many genes, and that we've shown a chromosome containing so many genes. So for example, this is a gene, okay, so it's a segment of DNA. Now let us discuss about what is a gene. How will you define a gene? A gene is defined in classical biological sense as a portion of chromosome. Okay, so it's a portion of chromosome. It's a part of a chromosome which determines or specifies a single character, or you can say a trait. Right. The gene can also be defined as a polynucleotide chain that contains segments of each containing or each controlling a particular trait. Okay. So, gene can be also defined as a polynucleotide chain containing segments of DNA which control a particular trait, a particular function. The gene is a sequence of DNA which is responsible for hereditary transmission from one generation to another generation. Okay, so genes are responsible for a transmission of hereditary characters. The gene can also be considered as a unit of recombination, that is recon. It can also be considered as a unit of mutation, that is muton, and a unit of function, that is cistron. And these three units of genes, that is recon, muton, and cistron, are not identical. So let us discuss about the units of the gene. Now, what are the various units of gene? Number one, recon. Recon is the smallest unit of gene capable of undergoing crossing over and recombination. Okay, it is the smallest unit of gene capable of undergoing recombination. Okay, where crossing over can occur, that means gene from one segment can be passed to another segment. Okay, and that is called as crossing over. Exchange of genes may take place, and that leads to recombination. It was thought that genes are part of chromosomes and crossing over occurs between two genes, rather you can say two different genes. Okay. However, it was supposed that crossing over may not be taking place within the gene. Okay. So earlier, what is a recon? It is the smallest unit of gene capable of undergoing crossing over and recombination. But then it was thought by scientists that crossing over genes are present on chromosomes, are parts of chromosomes, and crossing over takes place between two different genes. It was thought that recombination does not take place within the same gene. Okay, the gene was thought to be an invisible unit of a chromosome which controls some phenotypic characters. However, Benzer, on the basis of test for recombinations, showed that, okay, on the basis of test for recombination, Benzer, the scientist named as Benzer, showed that in viral DNA strands, crossing over takes place between the genes as well as within the gene. Okay, so recombination not only takes place between two different genes but also within the gene. One such subunit of gene where recombination can take place within the gene is called as recon. So what is recon? The smallest unit of gene capable of recombining genetically. What is recon? Smallest unit of gene capable of recombining genetically. The recon structurally consists of one or two pair of nucleotides. Okay, one or two pairs of nucleotides, possibly only one pair. Okay, so for example, if this is one segment of gene, right, and then what can happen? Crossing over can take place between two genes. That means there may be exchange of gene segments. Okay, so that leads to recombination within a gene. So that is about recon. Now the next unit of gene is 1 minute. Yeah. The next unit is muton. What is a muton? The smallest unit of gene capable of undergoing mutation. That unit of gene which is capable of undergoing mutation is called as a muton. What is a mutation? Sudden heritable change in the genetic makeup. What is a mutation? Sudden heritable change in the genetic, you can say, segment of a gene at genetic level. The muton consists of many pairs of nucleotides within the DNA molecule. Muton consists of many pairs. Okay. Now, what is muton? The smallest unit of gene capable of undergoing mutation. Okay, that is called as muton. What is mutation? A sudden heritable change in the genetic makeup of an organism. Now, what does a muton consist of? Many pairs of nucleotides within the DNA molecule. During mutation, changes at nucleotide level are possible. That means one base may be substituted or replaced by another base. Maybe one base may be deleted, one base may be added. So changes at nucleotide level occur during mutation. Okay. So what is muton? The smallest unit of gene capable of undergoing mutation. Now then third, you can say, segment of gene is a cistron. That part of a gene specifying a single polypeptide chain is called as a cistron. That means cistron is that region of the gene, that part of the gene which performs certain functions. And what are the functions? Gene is identified by its functional product, that is a protein or maybe an enzyme, right? So that part of DNA specifying a single polypeptide chain is called as a cistron. Thus, the gene as a unit of function, is called as cistron. Now, according to one gene, one enzyme hypothesis, gene was considered to be the unit for coding the synthesis of a single enzyme. Okay, so according to one gene, one enzyme hypothesis, one gene is responsible for coding the synthesis of one enzyme, right? That means the gene was identified by its functional product, that is the enzyme. However, one gene, one enzyme hypothesis can be expressed in several ways. Like we can say that one gene codes for the synthesis of one mRNA, one gene directs the synthesis of one protein, or one gene directs the synthesis of one enzyme. So similarly, we can change it: one gene, one mRNA, or one gene, one protein, or one gene, one enzyme hypothesis, right? So we can express that one gene, one enzyme hypothesis in a different way. Now, so what is a cistron? Cistron is the functional unit of the gene. Now, according to Benzer, why we call it a cistron? Why we call that region of gene which performs certain function as cistron? It is called a cistron because it is a chromosome segment within which operate cistrons effect. Okay. Now in E. coli, the cistron may consist of about 1500 base pairs. Every time the cistron starts with an initiation codon and ends with a termination codon. That is why we say that it's the functional unit when it directs the synthesis of a protein. Okay, when it directs the synthesis of a polypeptide chain according to one gene, one polypeptide hypothesis. One gene is responsible for synthesizing one polypeptide chain, and when one gene is directing the synthesis of a polypeptide chain, there has to be a start codon and a termination codon. Okay, so every cistron starts with initiation codon where the protein synthesis starts and a termination codon where the protein synthesis stops. It has been found that hundreds of units of mutation, that is muton, and hundreds of units of recombination, that is recon, exist within each cistron. Okay, so within each cistron are present many number of recons and many number of mutons, and hence cistron is a very long segment of DNA. So therefore cistron occupies a much larger chromosomal length as compared to muton or recon. So recon is a very small part containing almost usually one base pair. Muton is also very small region where mutations take place. But what is a cistron? It is the functional unit. So the cistron is very long segment, much larger chromosomal, occupies much larger chromosomal length, and a cistron consists of several mutons and recons. Okay, so that is about the concept of gene and the small units of gene, that is cistron, muton and recon. In the next one lecture we will discuss about what is overlapping genes, what are gene within genes, and what are split genes. Okay, so that are the different types, variation you can observe. Okay, now see, according to this one gene, one enzyme hypothesis, one gene directs the synthesis of one protein or one enzyme. But now we are going to study overlapping genes, where we will come to know that this hypothesis may not hold true for smaller organisms like viruses, right? So in the next presentation we will discuss about overlapping genes.